Hello, and welcome to The Issue on the ASGCT Podcast Network. I'm your host, Emily Walsh Martin, and today I'm thrilled to be joined by Dr. Jim Burns, CEO of Ensoma. Today, he will take us through the breadth of technological elements that Ensoma is bringing to bear to treat rare disease and solid tumor oncology indications. Ensoma is currently in the clinic for chronic granulomatous disease and have recently announced that they have infused their first patient in this trial. Jim will take us through the elements of this technology and how the first trial may help patients with CGD, as well as inform future therapeutic opportunities in other indications with significant unmet medical need. But before we get started, I wanted to take a moment to talk about ASGCT membership. We're in full-on membership season at ASGCT, and now is the perfect time to join the society. By becoming a member for 2026, you'll receive an exclusive suite of benefits designed specifically for cell and gene therapy professionals like yourself. ASGCT members also save up to $550 on annual meeting registration, a benefit that exceeds the cost of membership itself. Learn more or join today at asgct.org/join. And now on with this episode. Okay, welcome to The Issue on the ASGCT Podcast Network. I'm here with Jim Burns, the CEO of Ensoma. So happy to have you here today, Jim.
Thank you, Emily, and it's good to see you again.
Yes. And I know we're going to get really deep into the science and the strategy going on at Ensoma, and I'm really excited to hear about it. But before we do that, one of the things that I know our audience is always interested to hear about is, people's career paths. And I know that you started off in biomedical engineering, so it'd be great to hear your path from that to now.
Yeah, sure. I'd be happy to do that. And I get that question a lot in terms of my background and how I got to where I am. And at some point, I should give a talk on what's a nice bioengineer doing in a place like this as a CEO. But you're right. I did start off as a bioengineer or a biomedical engineer. I actually have an undergraduate degree in biology. Okay. And a lot of people don't know that. And then I decided to go to graduate school in bioengineering. And the reason I wanted to do that was because I really like the translation of science into things that can actually help people. And so I thought as an engineer, as a biomedical engineer, I could do that. So I got my PhD in biomedical engineering and then did a postdoc in material science and engineering because I was working in drug delivery. Mm-hmm. And when I did my material science and engineering postdoc, I was still doing drug delivery, but then I also migrated into materials that are used in surgery for medical implants and so forth. Mm-hmm. And surprisingly, my first job after 2 years of my postdoc was at a biotech company. It was at Genzyme. Okay. And Genzyme, one of the original biotech companies, but they were very eclectic. They had their typical biotech type of research and product development they were doing. They became famous for enzyme replacement therapies, but they also had a surgical products group that was using naturally occurring biopolymers for tissue engineering, drug delivery, for surgical implants. And so I was hired to try to figure out how to develop products from the polymer that they were using, the biopolymer, which was hyaluronic acid. So I built the group up at Genzyme. This is the very early days of Genzyme. They had just gone public. And, that work ended up going very well. I was very proud of the fact that we got multiple products into the clinic and actually multiple approved products. And that was just a terrific experience for me. And eventually what happened was at Genzyme is they ended up with a small molecule group, a therapeutic polymers group, and I became responsible for those groups as well. So in some ways, I was responsible for research for everything that wasn't a biologic. So it was biomaterials, it was therapeutic polymers and small molecules. Eventually, we got acquired by Sanofi. So that was a long time ago, 2011. And I was thinking I would get fired. Why would Sanofi need a bioengineer doing what I was doing? But ended up, I became responsible for what was called the Boston R&D hub for Sanofi, and eventually the North America R&D hub for Sanofi. That was a terrific experience for me, and learned a lot from that. I also learned that what I really loved was being closer to actual product development. So I moved to a joint venture to become CEO of a joint venture between CRISPR and Bayer. And that's where you and I got to know each other because you were at CRISPR at that time, Emily.
Absolutely.
And that was my first exposure to in vivo genetic engineering, because that's what we were trying to do with CRISPR, is in
Right.
vivo gene editing. That eventually got rolled up into CRISPR Therapeutics. And I became CEO of a company in San Diego that was doing RNA targeted gene therapy. And the science was going great there. And it was a really tough financial environment. So eventually we had to close Locana, despite the fact that the science was going really well. And here I am at Ensoma because I actually was not done in terms of what I thought I
Yeah.
could do in terms of product development. And I have to say, Ensoma is just a terrific company, and wanted to try to get something across the finish line in terms of getting something into the clinic. And I have to say, I think we're doing a great job there. We're really excited about where things stand.
Absolutely, and it's funny. I'm so impressed that you figured out so early on in your sort of career trajectory that you wanted to do something applied. It took me a lot longer to sort of realize, oh wait, maybe I don't want to describe disease. I want to contribute to teams that are trying to actually treat disease. So I love that you sort of figured that out so early and have been pursuing it ever since. Which brings us to Ensoma. I'm very curious about the in vivo gene engineering platform there. Can you talk a little bit about it? Tell me, what differentiates you from others, and also what therapeutic areas are you pursuing right now?
Yeah, the one thing I hope people come away from here is that what differentiates us for our approach is that we're creating a durable source of any immune cell to treat disease. So how do we do that? We are doing in vivo genetic engineering of long-term hematopoietic stem cells. And by genetically engineering those stem cells in the periphery, they go back to the bone marrow and they do what they normally do, which is they create more immune cells and blood cells. Except we've programmed these cells to create immune cells that are doing good. And I'll
Yeah.
explain what that might look like for a couple of the diseases that we're now working on. And I just want to say that this idea, which I think is absolutely brilliant, came from the Fred Hutch, from Hans-PeterKiem, as well as Andre Lieber at the University of Washington. This is their brainchild. And the company was founded in 2019 to take some of their research and to translate it into products that could actually help people. What really differentiates this platform is the fact that in modifying these HSCs, you have a durable source of these cells, just to be redundant, to treat disease. But the way we do it, the delivery system, is what's really key. That's part of the real secret sauce to this. And that's what Hans Peter, as well as Andre, had discovered in their labs at the University of Washington and the Fred Hutch.
Yeah, and it's always about delivery, right? I mean, it's funny. I liked hearing you say that you started your career at Genzyme thinking about delivery, and you're still doing it. But I think we all are doing that in the cell and gene therapy field. So, can you tell us a little bit about how you guys target the HSCs?
Yeah, and this was brilliant work that Hans Peter and Andre did, which they use what we call helper-dependent adenovirus. So it's a chimeric of an adenovirus that has a very high affinity for hematopoietic stem cells. And what Hans Peter discovered is that hematopoietic stem cells, especially long-term HSCs, express a high degree of CD46 on the surface. Most of our cells have CD46, but long-term HSCs have a lot of it.
Mm.
And so we've designed these virus-like, we call them virus-like particles or helper-dependent adenovirus. These have been designed so that they have a high affinity for binding to CD46. So we get, I wouldn't say only selective HSC binding, but we get a lot of binding of our viral particle to the HSCs.
Yeah.
And what that allows us to do is to avoid targeting tissues you don't want to. So, for example, the liver or
Yeah.
the spleen or other places, you get it to the long-term HSCs.
Yeah. No, that's super useful and super interesting. It's interesting, right? Like I think it feels like our field has been in a lot of ways, challenged with if you've got a disease where the liver is the impacted organ, we've got some ways to get some drugs there. But obviously, it sounds like one of the breakthroughs here is figuring out that CD46 based targeting can actually enrich or preferentially give you access to an entirely different compartment, those long-term HSCs, which is great. One of the, I guess, questions I had when I was first, you and I were first talking about this was sort of, how do you get to those cells? Because in a lot of ways, when I was first thinking about this approach, the long-term HSCs are kind of living there in the bone marrow, right? And tucked away. I imagine them tucked away, hidden from view. But you guys have a very clever approach to sort of overcome that tucked awayness of them.
Yeah, so they're in their niches in the bone marrow. And what we do is, and this is standard for transplant.
Right.
There's lots of diseases that are treated through stem cell transplant, and you have to mobilize the stem cells out of the bone marrow. And so we use a very standard way of doing that, which is we give a regimen of GCSF to the patient for a few days, and then we top it off with plerixafor, right,
Mm.
which binds to CXCR4. And so that helps release all these cells that you've induced to start to increase in number in the bone marrow. And that's how we mobilize the HSCs from the bone marrow. What's nice about this regimen with the GCSF as well as with the plerixafor is that it seems like it increases the number of long-term or primitive HSCs,
Mm.
which is exactly what we want for this therapy. So they get mobilized into the periphery, and then we infuse just with a single IV infusion our gene therapy product,
Yeah.
which then can transduce the HSCs. The other thing I should mention about this virus-like particle or the helper-dependent adenovirus is that, and this is very important for us, is it's really good at targeting and transducing HSCs, and it has a huge capacity. So
Right.
what's the capacity of AAV? It's about, what, 4.8 kilobases?
Yeah. And if you want to have it sort of looped back on itself, it's half that, right? So yeah,
Right.
you're very limited with an AAV.
Yeah, so the capacity of these virus-like particles is 35 kilobases.
Right.
So it's huge. So it allows us to do a lot of things in terms of multiplexing and doing a lot of different genetic engineering of the HSCs when we transduce them.
Absolutely. And so let's talk about one of the products that you're sort of pursuing with this platform, and it's the one that you're embarking on your first clinical trial with, and that's in chronic granulomatous disease. Hope I said that right. Can you share a bit about what this disease is for listeners who may not be as familiar with it?
Yes, and you did say it right, and it is a tongue twister.
It is.
I
I
still
feel
have
like it should
problems
have
with it.
another syllable in there. So,
So
yeah.
we just call it CGD.
Okay, perfect. Let's do that.
And so this is a primary immune deficiency. And what happens is that there's a mutation that occurs in the enzyme complex that's involved with creating reactive oxygen species in like neutrophils. So
Okay.
as you know, that's our first line of defense. We get a
Yeah.
fungal infection, we get a bacterial infection. The neutrophils are there first. They create oxygen-derived free radicals to kill the bacteria or the fungus. And so with this mutation, and there's many different mutations, there's different flavors of this,
Yeah.
they can't do that. And so patients at a very, very early age struggle with chronic infection, fungal
Okay.
infections, bacterial infections, and it has a lot of consequences. So obviously dealing with infection is tough, but then they also have uh, serious problems with granulomas, thus the name of the disease. And
Okay.
there's serious dysregulation of inflammatory processes that ends up creating inflammatory disorders like colitis.
Mm.
It used to be these patients would die very young,
Wow.
as children. Now, because of stem cell transplant, they live longer. So in Western countries, people can live to their 40s or 50s with this.
Okay.
The most severe form of this is an X-linked. It's X-linked CGD, and that's what we're working on. And what we're doing for this is, again, using our system, our delivery system, is that we are delivering a normal gene to the stem cells. Stem cells go back to the bone marrow, and then they expand, and we help with that. And they will create neutrophils that have the corrected gene in it.
Yeah.
Now, what's really cool about this is the fact that there's precedent that this works. There was a study done with an ex vivo therapy. So you take the
Sure.
CD34 positive cells, you roll them up, you transduce them in a petri dish. Yeah, I'm exaggerating. You
Yes.
put them back. This works really well, and they make neutrophils that are making the normal gene. And it had a profound effect on the patients that received this. So what we're doing basically is taking that ex vivo therapy and making it in vivo. We're taking basically the same gene that was successful ex vivo, putting it into our system, and we're putting it into stem cells, but we're doing it in the person. And then
Yeah.
those stem cells, make the product, the neutrophils that are normal.
And it strikes me it's kind of like you're taking the petri dish, as you put it, to oversimplify it, right? But you're taking the bioreactor out of the process, and you're basically doing all the very similar steps, right? So you mobilize the stem cells, which you would have to do if you were doing some sort of ex vivo therapy, but then instead of taking those cells out, transducing them externally and then putting them back in, you're basically treating them while they're in the periphery with the adenovirus approach. So it's very, very clever.
Yeah, that's a good way to put it, that the patient becomes their own factory
Exactly, exactly.
for this. Yeah.
And you talked a little bit about the fact that the payload size for this is much different than for AAV. It really opens up a lot of sort of opportunities, and I'm curious. I know one of the things that is part of your technology is that you want to make the transduction durable, right? And so you're using, I think, is it a Sleeping Beauty transposase to allow you to integrate your gene of interest into the cells permanently? That way, you're not dealing with the fact that the adenovirus might be an episome and could get lost over time. Is that how you're doing it?
Yeah, that's exactly right. So we're using a transposase system,
Yeah.
Sleeping Beauty. And what that does is it takes our gene and we put signals on either side of the gene that's in the vector, and the transposase will put that into the gene in the stem cell. It's a pretty efficient system. And what's nice about that is it allows us to insert large genes. And then, it's expressed, but it's expressed only in the cells that we want it to. This is really
Yeah.
important. So we have a neutrophil specific promoter.
Right.
So this gets expressed in neutrophils. It's not getting expressed in B cells or T cells or NK cells or erythrocytes. But what's really interesting about this is that we can see it in those cells. So it's there, but it's
Right.
not being expressed. And the reason why that's important for us is that we're also working in oncology. So we're using this exact same, or almost exactly the same system
Yeah.
for creating a multi-lineage approach for cancer, for solid tumors. So
Okay.
we can create a CAR macrophage, NK cell, and a T cell, CAR T,
Okay.
because we have the capacity to have a gene that large. And so that's what we're doing for solid tumor. But we can see this gene that we're putting into the neutrophils and expressing in the neutrophils. We can see that in M's, NKs, and T's. It's not expressed. So
Right.
this work with CGD, which is hopefully going to result in a potential cure for these patients, is
Right.
also establishing the platform's utility beyond primary immune deficiencies, but also for cancer, because we'll be able to see this gene in those cells for those M's and K's and T's. It just won't be expressed, but it'll show validation of the approach for solid tumor.
Absolutely.
Yeah. One last thing I'd like to mention is that, and we're really excited about this, is that we have infused our very first patient
Oh, that's great. That's
for
great to
CGD.
hear.
And this is the world's first time that an HSE has been transduced in vivo and a gene inserted for treatment of disease. So we're really excited about that.
That's amazing. Definitely looking forward to hearing more about that as the trial continues. So, okay, let's talk about actually some of the, what this may open up for you. Obviously, you're going to learn something in this first trial about the transduction efficiency, the if you will, genetic correction efficiency, which will correlate to an insertion efficiency, I guess, for oncology type implications. So tell us how you're thinking about indication selection for a platform like this once you have all of those sort of inputs sort of understood based on how this first trial goes.
So I love that question. And I can tell that you have a product development background because it's really important to think about what you're going to work on so that you have the most efficient and effective use of your resources for a new platform like this. And the way that we think about this, for example, with oncology, and the reason that we've gone into oncology is because number one is the scientific basis for your platform and your approach has credibility for treatment of the disease. The second is that you have an opportunity to, in a reasonable way, of actually showing that you're having an effect clinically. We all have limited resources these days for what you work on and how you
Yeah.
translate science into actual products. So you have to be able to show that you can translate your research into a product in a reasonable way clinically. But third is the impact. We have to show that this is going to have a substantial impact, unmet medical need. And I think that, for example, for CGD, we've shown I think we're going to show that. We can show that our scientific approach can work, that in the clinic in a reasonable period of time, and this is something that's going to have a substantial impact on this patient population. For solid tumor, so IO solid tumor, 90% of cancers are solid tumor.
Right.
There's so much more that could be done to help these patients. Well, what if you had an approach that allows you to bring the entire immune system to bear on solid tumor instead of just a single part of the immune system? That might have a tremendous impact. And that's what we bring to the table with our approach, a durable source of multiple parts of the immune system to fight a solid tumor cancer. And quite frankly, the opportunity economically from a business standpoint is very significant because there just isn't anything that does what our platform could possibly potentially do for solid tumor.
Yeah. And I mean, it strikes me, right, if I'm honest, in my career, while I've always had to think about the immune system, it is something that absolutely overwhelms me if I think about it too long, just because it is such a complex system of cellular interactions and handshakes and communication that has to go on. And so, to your point, the ability to have sort of a coordinate approach across different cell types that says, NK, listen up when you see a macrophage saying this to you. It really does sort of start to open up, in my mind, some really interesting biology, but some pretty complicated biology.
But you let the body figure it out.
Right.
So what we're doing is changing the HSC, in the case of solid tumor, putting in the instruction to make a CAR-M and a CAR-NK and a CAR-T.
Yeah.
But then the HSC is just making NK cells, Ts, and Ms like they normally would, macrophage or macrophages. And what's really interesting about this, and we have data to support this, it was recently presented at SITC. So the macrophages, our CAR-Ms, will go into the tumor, solid tumor, and it heats it up. Makes it more amenable for the NKs and Ts to come in and then kill the tumor cells.
Yeah.
And that's exactly what you want to have happen. You want the natural process for these cells to unfold, and that's what's the beauty of targeting the HSCs. And it's durable. These cells will be there and replenished to fight the cancer on an ongoing basis.
Yeah, which is something that I think, obviously, the jury is still out on sort of traditional CAR approaches in solid tumors. But one of the things that I think people are worried about is that durability. And so the fact that you are making a sort of a durable source of targeted cells seems to make a lot of sense. So very, very exciting. The one thing we didn't talk about, actually, is for therapies like this, sort of, I guess, dose projection, for lack of a better term, sort of how many cells do you need to correct, and sort of how do you get to that right number. There is one other thing that you're doing, at least in the context of your first program and in CGD, the EN374 program, which is trying to select for the cells that you've corrected. Could you talk a little bit about that as well?
Yeah, this is an important part of our platform. I think it's a real advantage. So we only have to deliver our vector to the cells once and just get an initial foothold. We don't need much. And so the nice thing about that, we don't have to overdose.
Right. Which again reduces the likelihood that it goes to the wrong cells. That by being able to have a much lower dose, presumably you can focus on those HSCs and maybe avoid the liver even more.
Yeah, exactly.
Yeah.
So, but what we engineer into our system is a way of selecting for our transduced cells over, I guess I'll call them the wild type cells for the patients.
Yeah.
So the transduced long-term HSCs, after we've infused our product, they go back to the bone marrow. We get an initial foothold, is what we call it. And what we've engineered into those cells is a mutant form of an enzyme that repairs DNA damage. And it works to repair the DNA damage. But what happens next is that we give these patients, let's say a month after the patient's been infused, a cocktail, I'll call it, of something called O6BG and temozolomide. Temozolomide is an alkylating agent. And what it does is it alkylates the DNA at a certain location in the stem cells, and then those stem cells die. Now, those cells have this normal form of that enzyme I was talking about. It's called MGMT, methylguanine methyltransferase, that will repair a certain number of those cells. what we do is that we put a mutant form of that into our cells. And so together with the temozolomide, we give O6BG, O6-benzylguanine, which is an analog of MGMT, and it basically blocks the ability of the MGMT to work.
Got
So
it.
it enhances the ability of the temozolomide to kill the wild type HSCs, and it protects our cells. So then they can expand. So this
Right.
is done in, I guess I would say, a gentle way so that there's some of the wild type cells that
Right.
are killed, and then our cells can expand. And that's how we actually control the dose to a therapeutic level. So we can do that one time, two times, three times. In our current clinical trial, we're doing it three, and we'll learn how many
Okay.
times that we need to do it.
Oh, that's interesting. Okay. So it's sort of a gentle enrichment for and a selection against, I guess, enrichment for the cells that received and that got transduced by the adenovirus and a selection against the ones that didn't. And in that way, you can sort of level out to the level that you think will be therapeutic in terms of the number of circulating neutrophils you have to eventually correct. Yeah. So fascinating. And I just have to say, obviously, what impresses me most, I think, about the Ensoma technology is the fact that you guys have had to solve a lot of different challenges and a lot of different technological obstacles to bring everything together to be able to have what could be a very flexible set of components to sort of address the next, set of indications that you decide to pursue. I'm curious a little bit about how do you move so many things forward in parallel and have them all come to bear so that you can, make some real progress in the clinic?
Well, one of the interesting things is that this system is relatively modular. And we've been spending a lot of time on optimizing the platform itself. And now what we can do is, I don't, this is an oversimplification, but it's almost like cut and paste.
Yeah.
So I'm thinking about CGD. That's basically the same system that we're going to be, and we are using for solid tumor.
Right.
All we're doing is changing the gene that we're putting in.
Yeah.
And,
And the
and there might be the
promoters.
promoters.
Yeah.
Yeah, because I
Yeah.
didn't mention the fact that we have macrophage specific promoters, myeloid specific promoters, NKT promoter. And so there's that, but it's pretty much the same system.
Yeah.
What's really nice about this, so we can basically take any stem cell transplant that's been successful before and take that and make it a product. And we can adapt that system for other diseases, as I just described for solid tumor. The other thing that's really important for this we haven't talked about is manufacturing. This
Mm.
is actually something that is very scalable. We can scale this up to thousands and thousands of liters. There's a tremendous efficiency. If you can make an monoclonal antibody, you can make this. This isn't like making an AAV gene therapy.
Right.
So that's the other part of this that's really, I think, important is the
Absolutely.
modularity of the manufacturing process.
Yeah. No, so many directions that you guys can go in now that you sort of got your components, which is fantastic, and it'll be really exciting for all of us to sort of watch your success as you progress in the clinic. Thank you so much for taking some time to talk about all the different components and walk us through the technology. We're at the end of our time, which means I get to ask my favorite question, which is if you could wave a magic wand, and fix one thing for cell and gene therapy, what would it be for you?
Well, we know that they're very effective. I mean, that's just growing evidence that cell therapy, gene therapy can really work. The business model is still a tough business model.
Mm,
If you're doing, that's why ex vivo cell therapies are now going in vivo. And gene therapies, they have challenges as well from a business model standpoint. I think we're going to get there, but that would be my magic wand is if we could ensure that the business model is one that's going to allow us to develop products that for whether it's a rare disease or somebody has limited resources, that they could get it. That would be my dream for a magic quantum
Absolutely.
moment.
Absolutely. Well, I absolutely hope that that comes true because I think you're absolutely right. I think if we don't have that sort of fullness of the, from discovery to actually, treated patients, reimbursed treated patients, then it's really hard to keep the momentum that I think the field has right now.
Well, Emily, I want to thank you for the opportunity. This has been great to see you again and have this conversation. It's really been enjoyable.
Absolutely, Jim. It is always a pleasure to chat with you, and thank you so much for sharing all your experience and your expertise and the progress so far at Ensoma.
Yeah, you're welcome.